Hi, I'm Jim Luby, Professor of Horticultural Science at the University of Minnesota, and along with my colleague David Bedford, we're both pictured in the photo on the slide, we've been leading the University of Minnesota apple breeding program. In today's presentation, I want to talk a little bit about the history of the apple and our University of Minnesota fruit breeding program, some of the challenges of apple breeding, and some of the features then of our University of Minnesota apple breeding program. First, let's talk a little bit about the history of the apple. Now, domesticated apples are not native to North America. Their origin is thought to be in Central Asia, what is now Eastern Kazakhstan and Western China. You can see them in the slide below. Most of the trees growing in the mountains there are apple trees in Kazakhstan. Now, the apple is thought to have traveled to Europe on trade routes that we sometimes know of as the Silk Road. It was grown in Europe for many centuries and then eventually made its way to America with early European settlers. And we had people such as Johnny Appleseed, Jonathan Chapman, and other nursery growers propagate apple trees as it moved across Eastern and Midwestern North America. Eventually, apples came to Minnesota with settlers from the East or from Europe. However, they found that the varieties they brought with them had a big problem. They didn't survive our Minnesota winters. In fact, a prominent newspaper publisher of the time, Horace Greeley, traveled to Minnesota in 1860 and said, "I would not choose to live in Minnesota because one cannot grow apples there." And indeed, fruits were a very valued part of the farmstead in what was then a very agrarian society in Minnesota. And so very desired. And the frustration of many people, many Minnesotans, that they could not grow apples and other fruits led to efforts to develop fruit breeding at the University of Minnesota. Now this started, the interest in this started with some successes that various citizens had, such as Peter Gideon, pictured on the left, who developed the wealthy apple in the late 1800s. It became very widely grown, not just in Minnesota, but across the northern US, named after his wife, Wealthy. And that led Minnesotans to think that more and better fruits could be developed for growing in Minnesota. And they petitioned the legislature to start a fruit breeding program. And that started in 1878 with Peter Gideon as the first breeder. That went for about a decade or so. It was discontinued for a while, but then in 1908, a new farm was bought where we currently have the fruit breeding program, about 35 miles west of the city of Minneapolis. And ever since 1908, fruit breeding has been going on at this site. It was initially called the University of Minnesota Fruit Breeding Farm. It's now called the University of Minnesota Horticultural Research Center at the Minnesota Landscape Arboretum. And pictured on the right is the first superintendent of that farm in 1908, Mr. Charles Harrelson, after whom the Harrelson apple was named. And that was one of the first new varieties of apples introduced from the young breeding program back in 1922. Since that time, there have been well over 100 new fruit varieties introduced, not only in apples, but in various other tree fruits and berries, and you can see them listed here on this slide. So let's talk about apple breeding. First, a few things to cover about the nature of apples and apple varieties. So apple varieties, or cultivars as we call them, are asexually propagated by grafting onto a rootstock. Now, asexually propagated simply means they are cloned, or we make exact copies of them by grafting them onto a rootstock. There's no sexual process involved. Why is this done? Well, it's an excellent way to reproduce a superior individual. You simply make exact clones of it. It's a bit expensive, but the market will pay for the higher cost of grafted plants rather than plants grown from seed, because apples are such long-lived plants. And then finally, there are actually some serious barriers to seed reproduction in terms of propagating a superior individual. First of all, apple trees grown from seed have a very long juvenile phase. Now, the juvenile phase is a period in apple seedling's life, much like in mammals, where an individual is incapable of fruiting or sexual reproduction. So that's one problem. The other one is one that many gardeners know. Apple trees are self-incompatible. They need another variety to pollinate and fertilize them. And then finally, apples are what we call genetically heterozygous, meaning they're not true breeding from seed. And so if you simply plant seeds from a variety, you're getting only half of the genes that come from the mother. The father will be something else, again, because of self-incompatibility. So another reason apple breeding is a challenge is because quality is so important in apples. In many of our field crops, of course, yield is really the important factor. But for profitability in growing and supplying apples, we need both high yield and high quality. And there are multiple quality traits that are essential for economic sustainability for a successful variety. I like to say that horticultural food crops like the apple are tasty, brightly colored, biologically active packages of water. That's why we like them. They're tasty, they're attractive, brightly colored, but they are biologically active, and that means they're subject to decay very readily if they're not handled right. And so quality and yield are really dynamic in produce, including apples. So what is quality in an apple? Well, first of all, there's how it looks, its color, size, shape, and probably even more importantly, how it eats, the flavor, the texture. And then there are other factors such as nutrient levels, and quality needs to be maintained with integrity during refrigerated storage. Apples are harvested basically once a year, and we like to enjoy them for several months. So they need to maintain their quality in storage. There's also sometimes a need for suitability for value added, such as for processing, let's say into cider or baked goods, and for different growing systems or eco-branding, such as use in organic production systems, where different options of growing are available. So the question then for apple breeders becomes what, how, and when to assess these quality traits. Assessing quality has to be in relationship to the quality that the consumer will encounter. So we have to consider factors like the stage of maturity, how mature the fruit is, when it was harvested, where it's grown, year-to-year variation in quality as we get warm years, cold years, wet years, dry years. Sampling fruits on a tree so that they're representative of the variety. How to store them, both the length of time in refrigerated storage and the conditions such as temperature, and whether we alter the atmosphere, such as reducing oxygen in storage to put the apples in a lower respiratory state. And finally, if the apples are going to be processed, somebody's got to do the processing and evaluate the processed product. So we have some tools that we use to help assess quality in terms of instrumental evaluation. For sugars, we use a device called a refractometer, which is pictured on the left, to look at acidity, which greatly influences our impression of both tartness and sweetness. We can look at the amount of acid by doing titration of juice samples with a titrator that's pictured in the center. And then for the texture of the apple, we can use a penetrometer, such as that one shown on the right, that can measure the hardness or firmness of an apple. But what it really comes down to eventually is what we call sensory evaluation. Our primary tool as humans. Although we have instrumental evaluations we really have to harvest and evaluate at proper maturity with our own human senses. Our sense of taste, our sense of smell, and our textural senses. So the perennial nature, perenniality of the apple tree is another challenge that we face in breeding apples. Like other woody plants, it has a very long life cycle and the juvenile phase that I mentioned previously. Sometimes it can take 10 years from germination to flowering. So it's a long time to wait. And plus, apples are big plants, so a lot of space is needed to grow them. And that's why apple breeding programs generally need a lot of space and a lot of time to develop new varieties. Genetics is another reason that apple breeding is a challenge. Apples, as I mentioned, are self-incompatible and they're genetically heterozygous. What that means is that it's difficult to predict offspring performance even when crossing the best parents. So about apple genetics, we can say two things about apple seedlings. First of all, none of them will be exactly the same as their parents, and no two are identical, even though they have the same parents. This is much like humans, in fact, where no child is exactly like either one of the parents, and no two children are exactly the same. And in apples, we don't even have such a thing as identical twins. Another reason apple breeding is a challenge is there are a lot of traits needed for a successful variety. We've got a number of fruit traits, for example, related to fruit texture, such as crispness, juiciness, and firmness, and also flavor, such as sweetness, acidity, and the aroma. And appearance, such as the color and the finish or brightness of the fruit, the shape of the fruit, the size of the fruit, and then certainly the ability of the fruit to store for some period of time and present good quality out of storage. But it's not just fruit traits. There's also a number of tree traits that are needed, such as disease resistance, for example, to apple scab, and fire blight, which are our two most important diseases in the Midwest. Productivity, certainly. Annual bearing, that is bearing a crop every year, rather than biennial bearing, which some varieties will do, that is having a crop only every other year. Premature fruit drop before the fruit are ripe is certainly something we don't desire. And then finally, tree form, a tree that's relatively easy to manage. So our breeding priorities in the University of Minnesota breeding program are what we call both market-driven and production-driven. We think about, first of all, why does a consumer purchase an apple or want to eat an apple? And then we can't just consider things that the consumer likes. They've got to be produced in our climate efficiently. And so we've got to consider some production-driven factors as well. Now, most important for us is the texture and flavor of an apple. These are the factors that make a consumer want to repurchase and eat a particular variety again and again. Appearance is also important though, because it's an important factor in the initial purchase of an apple. And then the pack out, which is the percentage of fruit that are fit to go to market, the storage in refrigerated storage, and the shelf life, that is the life of the fruit after it reaches the supermarket shelf or perhaps on the table of the consumer, have to be considered as well. The appearance, the texture, and the flavor have to be excellent throughout storage and throughout the supply chain till it gets to the mouth of the consumer. So now we're going to, in the next few slides, look at the various steps in our breeding program, which takes about 20 to 30 years from crossing to getting new varieties to the supermarket shelf. The first step is to choose the parents that we're going to use and pair them up and then make that cross. The second step is to test the hybrid seedling offspring from those crosses. The third step is what we call the second test, and that's a test of cloned plants of the selected individuals from the first test. Then there's the third test, which is a test under commercial conditions of the very best selections as we get closer to introduction and commercialization. And then finally, there are a number of steps that we have to take as we introduce a new variety, such as virus indexing of mother plant trees, intellectual property protection and licensing, and then nursery propagation. So that crossing phase, we'll start with that. That begins really in the winter and early spring when it's still pretty snowy in Minnesota. We compile the data for each potential parent we want to use, and this is both performance data and DNA data, or data from DNA tests that predict some of the traits that the parents will carry. And you can see us doing that in the photo here. And parents are paired then to complement their strengths and weaknesses. So we might have one parent that's disease resistant but not crisp. We would cross it with another parent that's maybe susceptible to a disease but very crisp. Then the pollination occurs in the spring, usually in May. It starts with covering the flowers before they open with wax paper bags. When the flowers are open and ready for breeding, the bags are briefly removed. Meanwhile, we've collected pollen from another tree that we want to use as the father, and that pollen is in the Petri plate, as you see in the larger photo. The breeder will use some of that pollen and hand pollinate each flower that was in the bag with his or her fingers, as you can see in the photo here. And eventually that pollen will hopefully germinate and fertilize the eggs in that flower on the mother tree. The flowers are then covered again after pollination, after we pollinate them, to prevent further pollination by bees. And then they go through the growing season. And this process produces fruit containing hybrid seeds inside. So each seed has genes from both parents. It gets genes from the mother tree where the cross was made, and then half of its genes also from the father tree where we collected the pollen. Now, the pollen source doesn't affect the fruit, just the seeds. We get this question all the time. If my Honeycrisp tree is pollinated by a crabapple, will I have crabapples on my Honeycrisp tree? No, the fruit is maternal tissue, it's Honeycrisp tissue. Only the seeds are carrying the genetic contribution from the pollen parent. And of course, that's exactly what we want in breeding. So those fruits are collected in the fall, the seeds are removed from the fruits and given a 60 to 90 day cold stratification that simulates what they would go through in the winter. They're placed in moist sphagnum in a refrigerator at just above freezing. And then they're germinated, usually in January or February, and the young plants are grown in pots until the end of July. Now, during that time, we do our DNA tests to cull, or basically get rid of, undesirable seedlings. In this process, a small leaf is collected from each seedling and sent to a laboratory for DNA tests that predict its important traits such as fruit texture, storageability, and disease resistance. And those seedlings that are predicted to be inferior for some of these traits are discarded before they take up room in the seedling orchard. So now we'll move on and talk about that first test of those hybrid seedlings. So during this first test, each seedling is propagated onto a dwarf rootstock. 2 buds are collected from each seedling and grafted by budding onto a dwarfing rootstock at very high density, as you can see in the photo, about 3,000 trees per acre. And these are very short dwarf trees supported by a trellis, and you can see some of the trellis posts there. Then we have to grow these seedling trees. They're maintained in place until they begin bearing fruit. Usually we have about 20 to 25,000 trees planted at any one time. And you can see the breeders here going through a seedling orchard as it's starting to bear fruit, usually after about 4 or 5 years of growing the trees. And that's when the real work begins, between about years 5 to 10. Each seedling has to be tested. Tasting can involve up to 500 apples per day. Now, of course, we don't swallow that much, but We still have to bite them, crunch them, and taste them. So it's a big job to do this sensory evaluation. That's our primary tool, as I mentioned, for quality assessment in apple breeding. We have to evaluate each seedling tree. Now, I mentioned we have about 20 to 25,000 seedlings in the field at any one time. Usually each year, about 6,000 to 8,000 of those are fruiting. And so the fruit has to be tasted by the breeder in the field at the proper ripeness. So it's actually got to be checked about every week for about 1 to 3 weeks, and then tasted when it's ready. And so that process can, as I mentioned, involve tasting up to 500 apples per day. So it's quite a load during those peak times in September. Now, only the best trees of those thousands of individuals in first test are selected for further testing. Most of them are discarded during the first year of fruit bearing. And only about 1 tree out of 200, or about 0.5%, is selected to go on for further testing. We call these selections in the breeding program. Each selection is assigned a unique test number. For example, Minnesota 1711 was the test number for the variety that became Honeycrisp, and Minnesota 1914 for the one that became the apples we know as Sweet Tango apples. The fruit is stored and evaluated over the winter from these superior trees or selections. And then each of these individuals is cloned by grafting for the second test stage. So that's again shown in this schematic. The second test is where we test clone plants of the selections. So first they have to be propagated or clonally reproduced. By grafting, we use a technique called budding during the late summer, which is shown in the photos below. 2 trees of each selection are propagated on M26 and B9 rootstocks, which are common rootstocks used in the industry. And then these trees are grown in our second test block. In this second test, the trees of selections are evaluated for another usually 10 to maybe 15 years. And they're evaluated for about 25 tree and fruit characteristics. So it's a pretty thorough testing. It might be cold hardiness. You see a tree injured there in the inset photo, and then a number of fruit characteristics. And during that time, most of them proved to be inferior for one or more of these characteristics. So only the best selections are propagated and sent on for other sites in what we call a third test. So this again shows our schematic, and then we go from the second test to the third test, which is what we call our pre-commercial test. The trees are grown under commercial conditions, finally hopefully leading to introduction and commercialization of a new cultivar. So in this third test, or pre-commercial test, it's usually about 15 to 20 years after the cross was initially made. We'll propagate those best selections from the second test for commercial testing. They're sent out to commercial growers in Minnesota and across the United States, as well as other research stations, to get more opinions of whether they might be suitable to eventually be introduced as a new variety. If we get good reports and there's interest from growers, we go through the commercialization process, and trees become available to producers, usually by about year 20 to 25. The fruit producers have to grow those trees, and finally, a first substantial amount of fruit is available to consumers, usually about 25 to 30 years after we make the crosses. So these final stages leading to commercial production, as I mentioned, can take considerable time. I've got the last 5 introductions here listed from the time from crossing to introduction. Honeycrisp, for example, took 30 years. In more recent times, with varieties like Sweet Tango and First Kiss or Rave, it's been more like 17, 18 years. So it's gradually improving, or getting shorter, due to some of the improvements that we've made in testing processes. And we hope it will continue to improve and bring new varieties of Minnesota apples to you. And so I want to thank you and encourage you to enjoy eating University of Minnesota apples.